I'm off. So today we're going to have a lecture on drugs, microbes, hosts, the element of chemotherapy. So on last week's lecture we started talking about how do we physically kill or remove potential problematic microbes. And this was primarily dealing with microbes on surfaces, so things like instruments and tables and desks and so forth. Today we're going to focus on how do we physically remove these from living tissues as well as complete organisms such as ourselves. So we talked about this before pretty early on in the semester, but the goal of all antimicrobial therapies, chemotherapies, is to essentially remove the microorganism of interest while leaving our cells intact. So this is this concept of selective cytotoxicity, or targeting only the organisms of interest and leaving our cells alone. Now, it sounds relatively simple, but it can be very, very problematic and complicated, especially when you're dealing with things like viruses, and we'll talk about this in a little bit. But not only do you only want to target microorganisms, you also only want to target the microorganism of interest. Remember, there are many commensal organisms found throughout our body, and if we use a broad spectrum chemotherapeutic agent that targets all microorganisms, we can potentially harm these beneficial organisms or commensals, and this could do more harm than good in the long run. So it's this kind of fine balance between targeting only the pathogenic organism while leaving our cells as well as our commensal prokaryotic organisms alone. And as you can imagine, this can become very, very difficult. So some characteristics of an ideal antimicrobial drug. So obviously you want them to be selective toxic to the microbe, but non-toxic to host cells, as well as our commensal cells, or prokaryotes. You want it to be microbial-cidal, so it kills the organism rather than just being microbial static or preventing growth. You want it to be relatively soluble. The more soluble, the easier it is to get into tissues as well as the blood. Remain potent for long enough to act, so you want it to have a relatively long half-life. This is one of the reasons why people use things like erythromycin, is because it lasts for around 72 hours in the blood, so you only have to take it for 5 treatments instead of something like amoxicillin, which requires about 30 treatments. You want it to remain potent for long enough to act, and it's not broken down or excreted prematurely. Most drugs are seen as toxins once they enter the body, so our livers and kidneys will try to filter them out and process them. So if we can develop a way of administering a drug so that this doesn't happen, or if it does happen, it actually makes the drug stronger, it can greatly increase the efficacy of the drug. It does not lead to the development of antimicrobial resistance, so obviously you want the drug to work relatively well on all of the microorganisms of interest. If you leave a few of them behind, it obviously can result in resistance, and we'll talk about this concept later on. You want it to complement the host defenses, so this is a big point here. Clearly you want it to work against the microbe, but you also want it to work with the body as well. And by work I mean not target the body, instead complement the natural host immune system. If you have a drug that will kill the immune system, but it kills the targeted organism as well, it's not going to work at all. All of these drugs need to work and complement with the immune system for them to function properly. You want it to remain active in tissues and bodily fluids. So usually these drugs work fairly well in bodily fluids such as blood, but it can be harder for them to penetrate things like tissues. And this is why deep tissue infections such as MRSA or VRSA can be very, very difficult to treat. Not only are these organisms resistant to many antibiotics, it's also difficult to get the antibiotic physically into these tissues. You want it to be delivered to the site of infection. You want it to be reasonably priced, and most of these aren't, and does not the host's health by causing allergies or predisposing the host to other infections. So clearly, you're introducing something into the body that is not normal, and occasionally this can have some severe interactions with the body's immune system, resulting in anaphylaxis or severe allergies. And we'll talk about this a little later on. So here are some definitions, which are pretty important to know. I'll talk briefly about them now because I will be using these words fairly interchangeably throughout. So chemotherapeutic drug, this is any chemical used to treat, relief, or prophylaxis of a disease. And by prophylaxis, I mean preventing the infection from occurring. So you take the drug usually in a lower dose, and this will prevent the organism from actually setting up shop within the body. Antimicrobial chemotherapy is a chemotherapeutic that is targeted against a microbe. And an antimicrobial is essentially any drug in this class. So whether it's naturally found in the environment or it's synthetic, it is just something that targets microbes. An antibiotic, which is the most commonly used term but isn't always used appropriately, is essentially a drug that is natural in origin, so it has come from another organism that is useful in targeting microorganisms. A semi-synthetic drug is essentially an antibiotic that we have chemically altered to make it more effective. So it has a natural origin, it comes from another organism. However, we are slightly modified so it works better. A synthetic drug is something completely designed and developed in the lab. It has no natural component or compound found in the environment. And these tend to be the most expensive because they require the most R&D or research and development. And a narrow spectrum drug is a drug that works on just a targeted group or type of organism, where a broad spectrum drug tends to be wide encompassing and target many, many different types of groups or flavors of organism. So antimicrobial drugs have been around for about 70 years now, first discovered in the 40s, and they were discovered by isolating fungal compounds, primarily fungal compounds, that are used to inhibit or kill bacterial foes or enemies. So the first one I'm sure most of you have heard about is penicillin, which is from the Penicillium mold. And this is a compound produced by the mold to kind of inhibit bacterial growth, since most bacteria from molds are competitors, so they're going after the same limited food sources. So if the mold can target these bacterial competitors, they open up the food sources, making them more available, and allows them to grow better. So penicillin was discovered again in the 40s by Alexander Fleming. It's kind of a happenstance discovery. He was a pretty dirty microbiologist, and he had some fungal contamination on some of his bacterial cultures, and he noticed that these fungal contaminations inhibited bacterial growth. He then played around with the fungus a little bit, found the actual compound, penicillin, which was doing this, and used it to treat clinical cases. It worked fairly well. However, penicillin has some major problems, and we'll talk about those in a little bit. So again, the goal of antimicrobial drugs is to disrupt the cell, processes, structures of bacteria, fungi, and protozoa. And again, you want this to be selective just for these groups so that it does not affect our cells. Or it can inhibit viral replication, and this is primarily dealing with viruses, which can be much, much more difficult to design drugs to, and we'll talk about why in a little bit. Most of these will interfere with the function of an enzyme, which is required for synthesis, since enzymes, like we've talked about before, are very, very specific. Typically, if you target an enzyme that is required early on in the enzymatic pathway, it will affect everything downstream of that. And if that's an important enzymatic pathway, it can kill the cell. And again, they should be selectively toxic, which means they kill the microbial cells while leaving our cells alone. So some types of antimicrobials, the big groups here are the cell wall inhibitors. So this is primarily targeting peptidoglycan, but it can also target mycolic acid synthesis. And we talked about this before. Essentially, microbes need to constantly be producing their cell walls. If they do not, their cell walls will weaken and they will essentially explode. There are some drugs that are drug classes that will target cell membrane. However, these rarely are used internally because they are not very specific to microbes alone, so they tend to do damage to our cell membranes as well. There are also a few drugs that will target DNA-RNA synthesis. However, this isn't a very common class just because this mechanism is what we call highly conserved. This means that essentially in microbes, the mechanism used for DNA or RNA replication or transcription is essentially identical to what is found in our cells. So if you target it in microbes, you'll probably target it in our cells as well. The big group here, number 4, this is the protein synthesis inhibitors, and these are the inhibitors that target the ribosome. So we've talked about this before, that the ribosome in prokaryotes is pretty different than the ribosome in eukaryotes. And the ribosome is essential for life because it is used for translation or the production of proteins. So essentially, if you can target the ribosome, you can also inhibit the growth and also potentially kill the microbe. And there are many, many drugs in this class, and this is where most drugs are currently being developed. But we'll talk about some caveats to this later on. And then the last group, which is not very effective, but it does work in some cases, is the folic acid synthesis pathway. Microbes need to produce their own folic acid, which our cells do not. So if you target this pathway, you can prevent the organism from growing and eventually kill it. So mechanism of drug action, so this is just the other groups that we've talked about in that picture slide. So cell wall, nucleic acid, protein synthesis, cell membrane structure, function, and or folic acid synthesis. So targeting the cell wall, again, active cells, so cells that are living, vegetative, not endospores, need to produce cell wall almost constantly. And this cell wall is important, especially in gram-positive bacteria, and this prevents the organism from essentially exploding. Now, as you would imagine, most of these drugs will not work very well with gram-negatives, and again, this is because they have that thick outer membrane which kind of takes over for the thin peptidoglycan cell wall in gram-negatives if it is removed. Some groups or some specific drugs in this class. Penicillins, cephalosporins, these react with one or more of the enzymes required to complete the process of production of primarily cross-linking or producing long strands of peptidoglycan, which can be used to produce the cell wall. And these are all bactericidal. They do kill the organism. They do not just stop its replication. So this is just a nice little cartoon showing what happens when you remove the peptidoglycan cell wall. Essentially the cell explodes because it no longer has that nice external rigid structure. So here you can see what a normal cross-linked peptidoglycan cell wall looks like. It has these nice little green dots, which for our purposes just means they're forming nice sticky bits that connect these long strands of peptidoglycan together. And when you remove this cross-linking, you essentially get just a whole bunch of strings of peptidoglycan which can no longer keep the cell together, and it will eventually explode. Other drugs that target cell wall, so bacitracin, this is for or from Bacillus subtilis. This is actually another prokaryote or bacteria that produces this antibiotic to inhibit competitive growth of other bacterial organisms. And obviously this bacitracin would not work with Bacillus subtilis, but it does work well with many other types of bacteria. And you can find this in things like Neosporin. So Neosporin is a topical antibiotic cream. Isoniazid, which is a TB drug, this will target the production of mycolic acid. It works relatively okay, but it does have some high cytotoxicity in our cells. And with all TB treatments, you never treat with just one drug. Usually you have to treat with 3, so triple therapy, and we'll talk about some of those later on. And then vancomycin works fairly well. It's probably the best drug in this class, and it works well for people who have MRSA. because MRSA is methicillin resistant, not vancomycin resistant. However, there is a new flavor of resistant Staphylococcus aureus called Versa, which is vancomycin resistant as well, and this can be one of the most difficult organisms to treat. And vancomycin also works well for people who have penicillin allergies. Because penicillin is a naturally occurring compound, typically allergies are more common with these naturally occurring compounds, just because our immune system is designed to target naturally occurring compounds. So antimicrobial drugs that affect nucleic acid synthesis. So this primarily blocks the synthesis of nucleotides in both RNA and DNA. So it can inhibit replication of the DNA, which inhibits the cell from dividing, eventually killing it. Or it can stop transcription, which is the production of RNA molecules, and these RNA molecules are then used later on for translation and the production of protein. So if you stop the production of the RNA molecules, you thus stop the production of a protein. And again, this is all inhibiting DNA or RNA synthesis. Now, to prevent transcription of DNA, this is a fairly difficult target because, like I mentioned before, it is highly conserved between species. So what I mean by that is in prokaryotes, the mechanism used for DNA replication is very similar to the mechanism of DNA replication in eukaryotes or our cells. So if you target the enzymes in prokaryotes, you'll probably target the enzymes in eukaryotes, resulting in really nasty side effects or toxicities. However, there is one drug out there that has been shown to work, and this is rifampin. This binds to RNA polymerase. It binds primarily to prokaryotic RNA polymerase because it is slightly different than ours, but it does have a lot of cytotoxicity, so it's not used fairly often, except in the cases of tuberculosis. So this is another one of those triple therapy drugs used in tuberculosis, and this one has very high cytotoxicity or side effects. And for that reason, when you're on this drug, you need to go to the doctor fairly frequently to get checkups. It's specific for bacteria, prevents elongation of transcription after initiation, and useful drug for tuberculosis. DNA replication, again, this is a highly conserved mechanism, so it does lead to toxicity, so it's rarely used. However, there is a molecule called DNA gyrase, which isn't found in eukaryotes, and there is a target or a drug that can work against this nucleic acid, but it doesn't work extremely well. Fluoroquinolones also fit into this category, primarily ciprofloxacin, and this is used really effectively in anthrax infections, primarily pulmonary anthrax infections. Blocking protein synthesis. This is a very common class of antimicrobials. Probably about 60% of the drugs fit into this category. And this inhibits translation, and again, it's by usually binding to the ribosome and changing the ribosome so much that it can no longer transcribe. And again, prokaryotic ribosomes are fairly different than eukaryotic ribosomes, so this gives us this selective toxicity. So our ribosomes are left alone where theirs are targeted. And there are many different groups or classes of drugs that fit into this protein synthesis: aminoglycosides, Sulfonicols, Oxazolidinones, Tetracyclines, and Erythromycins. Disrupting the cell membrane function. So this is essentially breaking down the cell membrane. Most of these work as detergents or surfactants. And we talked about these a little bit last week, but they essentially bind to the membrane, making the membrane hydrophilic, which essentially results in the cell losing its ability of keeping its environment enclosed, and it basically dies or explodes. Specificity for this doesn't work extremely well. It typically targets most cell membranes, so they tend to be fairly toxic. So most of these drugs that distribute cell membrane function are what we call topical antibiotics, which means they are used on the surface of our skin, and this is because our skin is covered in keratinized dead skin cells, which aren't affected by this cell membrane disruption. So some membrane active drugs, they're primarily detergents. They bind to phospholipids or lipid A in the phospholipid bilayer or cell membrane. This disrupts the membrane. Again, it has very poor selective toxicity. They do work very well, but they will also work very well on our cells as well. Polymyxins is a very common drug in this group, and you'll find this in a lot of triple antibiotic creams, such as Neosporin. And it works pretty well, but again, you only want to use it topically. Inhibiting folic acid synthesis. So this is what we call competitive inhibition. This is by introducing a drug that will actually bind to an enzyme in the folic acid pathway, and it uses this enzyme up, preventing it from producing folic acid for the bacteria. And as we mentioned before, folic acid is required for bacteria, and they need to produce it themselves, unlike ourselves where we can get it from the environment. And this is a metabolic analog, which means it looks very similar to folic acid, but it does not end up as folic acid at the end of the day. So it blocks the enzyme's catalytic site or active site by using the enzyme up, but it doesn't give the bacteria its folic acid at the end of the day. So this is what it looks like. Essentially, sulfonamides or trimethoprim, these are all sulfa drugs, and they essentially will bind to the catalytic site or active site of the enzyme, and this will prevent the enzyme from manipulating PABA, the main substrate in the folic acid pathway, or excuse me, prevent the enzyme from modifying PABA, and this eventually prevents the production of folic acid. So now we'll talk about some of the antimicrobial drug classes. We're primarily going to talk about prokaryotic antimicrobials. We will talk about some antifungals, some antihelminthic, and antivirals as well at the end of the talk. There are around 260 different antimicrobial drugs available, and they are classified into 20 drug families. And the largest number of antimicrobial drugs are for bacterial infections, but a big problem we're noticing is that bacteria are fairly quick at developing resistance to a lot of these drugs, and unfortunately producing these drugs, at least from a pharmaceutical standpoint, isn't as cost effective, or doesn't make the pharmaceutical companies enough money to make it a viable option. So what we see happening now is that there are relatively few new antibiotic drugs or antimicrobial drugs coming out into the market, which means it's only a matter of time before we have a series of microbes that are completely resistant to all antimicrobial drugs available. So it's a little bit scary to think about, but it is something that are realizing this and are trying to make this a more viable option. So here are some of the classes, and on this chart what we see here are the different groups of drugs that we'll talk about, and then you'll notice that there's a bar above them, and this shows you what types or groups of organisms this drug targets. So you can see some of them are what we call broad spectrum, such as tetracyclines, and tetracyclines will target gram-negative, gram-positive, chlamydias, as well as rickettsial organisms. So it targets a very large group of organisms. Now this can be nice if you have an unknown infection. So say if you have a patient, he has some sort of severe systemic or septic infection, and you have absolutely no idea what it is. Well, clearly, it's very hard to just target that organism since you don't know if it's gram-positive, gram-negative, or whatever the case may be. So the best option would be just to treat with a very broad-spectrum drug like a tetracycline or carbapenems. And essentially, what this will do is it will target the organism that's causing the disease, but the problem with these is they tend to target commensal organisms as well. So when this happens, your commensal organisms will die off, and you tend to get fairly sick, primarily in the gastrointestinal tract where you get severe diarrhea. But it's better than dying from the septic infection. So typically we use these if we're kind of unsure of what the organism is, or if it's something outside of the body like topical treatment where it doesn't really matter, you just want to target as many organisms as possible. But then you can see some of these smaller guys like polymyxins or topramycins. These will target only a handful of organisms, or like a small group, such as gram-negatives. And these are what we call narrow-spectrum drugs. And these are really nice if you know exactly what the organism is that's causing the disease, and you know what type of organism it is, so that it's a gram-negative or a gram-positive. What this will do is it will target only those groups of organisms, leaving all the other ones alone. And this will help your commensals and prevent them from dying from the drug. So here we can see Microbacteria. So these are the primary organisms that cause tuberculosis. Have a relatively small group of drugs, so isoniazid, which we've talked about before, and streptomycins, which we talk about in a little bit. So the first group we're going to talk about are the cell wall inhibitors. So the first major class in this group are penicillins. And again, this was discovered by Alexander Fleming. It was the first antibiotic discovered. And this primarily works by blocking the cross-linking of peptidoglycan. So without the cross-linking of these peptidoglycan strings, essentially the strings just kind of fall apart and the bacteria will eventually explode. All penicillins have something called a beta-lactam ring. And if a bacteria wants to get around a penicillin, they will produce an enzyme called beta-lactamase. And what this does is cleave off this beta-lactam ring, making the penicillin essentially ineffective. They have different spectra of action. And again, penicillinases or beta-lactamases essentially are a way for the bacteria to get around these penicillins. And they can produce these enzymes fairly rapidly, so you can develop resistance to penicillins fairly quickly. And here are just some examples of different penicillins: methicillin, tarcocillin, cloxacillin, and carbonicillin. So you can see all of them have the cillin nomenclature, so it's pretty easy to determine it's a penicillin. Here are these R groups. And then you'll notice all of them have this nice beta-lactam ring. Cephalosporins, so these are kind of these are a semi-synthetic flavor of penicillins. And essentially they have a beta-lactam ring, just like you would find in penicillins. But instead of just having one R group, they essentially have two R groups, and these R groups are functionally different. What this does is it blocks the penicillinase or the beta-lactamase from binding to the beta-lactam ring, and this will prevent them from cleaving it and essentially degrading the antibiotic. So they work well. Most of the drugs that we currently use to treat gram-positive organisms fit into this cephalosporin group. And they all have different spectra of actions, but most of them work fairly robustly. So cephalosporins, they're broad spectrum, but again they primarily target gram-positives, and this is because gram-positives have the thick peptidoglycan cell wall that they are heavily reliant on, where gram-negatives do not, so these do not work very well against them. It works well if you have an organism that's resistant to penicillins or has a penicillinase, and that is because this drug does not get cleaved by the penicillin, so it works, remains effective. Causes fewer allergic reactions than penicillins, and this is because it is semi-synthetic, so typically your immune system does not recognize it as foreign as such, and it does not mount an immune response to it. And there are essentially 4 generations of cephalosporins that exist, and again they're based on their antibacterial activity. And each generation essentially is the different iterations of the drug. So the first generation tends to be the earliest flavor of the drug, and the later generations tend to be the more recently developed. Now we're going to talk about the drugs that target protein synthesis. So this is primarily targeting translation, or the ribosome. The first group are aminoglycosides, and this binds to the 30S, or the small subunit of the prokaryote. And this distorts the ribosome so that it can no longer bind the mRNA properly, and this causes translational errors. When you have translational errors, you get misformed proteins, and misformed proteins tend to be very bad and not effective. A couple examples of drugs in this class are streptomycin. Streptomycin works fairly well, but it has a pretty nasty side effect that can occur in about 1% of the individuals who take it, and this is ototoxicity. Ototoxicity is essentially toxicity that forms in the eardrum, and this can result in deafness or loss of hearing. So it's a pretty serious one, and it's irreversible. So it's something we keep in mind when you're trying to treat with the streptomycin drug. So typically streptomycin isn't used unless it is the only option left. Neomycin also fits into this group, and this is found in triple antibiotic creams such as Neosporin, and this works pretty well in that case. And you don't get the ototoxicity associated with it because it is used topically. So aminoglycoside drugs, most of these come from some flavor of organism, either Streptomyces or Micromonospora, which is a fungi. Both are fungi. They're relatively broad spectrum because they inhibit protein synthesis, and you can use them for gram-negatives, so that's a nice plus to these. Remember, gram-negatives can be hard to treat with things like penicillins or beta-lactams, and that is because they do not rely heavily on the cell wall. Can also work, streptomycin works well for bubonic plague and tularemia, and also it's a fairly good anti-tuberculosis agent, but again, this ototoxicity is an issue. And gentamicin is less toxic, and it's used for gram-negative rods. Tetracyclines, so this blocks the A site, which is one of the translation sites within the ribosome itself. And if you look here, you can see the A site. This is the E, the P, and the A site here, and the A has got this big X here because it's being blocked. It prevents tRNA entry, so without tRNA, you're not going to bring the amino acid. Without the amino acid, you're not going to form your protein amino acid strain or chain or peptide. It works fairly well, and it works pretty robustly against all classes of microorganisms except for Mycobacterium. So it's very broad spectrum, but it is reversible. So for that reason, it is what we call bacteriostatic. So it inhibits replication, doesn't necessarily prevent or kill the organism. However, if you use relatively high doses of a tetracycline, you can kill the organism. But at these high doses, it tends to be pretty toxic to ourselves as well. And because it is broad spectrum, it tends to target many commensal organisms as well. And when this happens, it can be pretty nasty, especially in the gastrointestinal tract for the patient, just because it's you're losing a lot of commensals that are used in digestion. So they're broad spectrum. They can cause GI problems in some. And the reason why it's only in some is because we all have relatively unique flora in the gut or organisms in the gut. Some of them are hurt more by the tetracyclines than others. And it works well against gram-positives, gram-negatives, rods and cocci, aerobic and anaerobic bacteria. You can use them for things like Microplasmas, Rickettsia, and spirochetes. Doxycycline is probably the most common oral flavor of tetracyclines. You can use it for a variety of drug or diseases. So sexually transmitted diseases, primarily chlamydia, Rocky Mountain spotted fever, Lyme disease, typhus, mycoplasma, pneumonia, cholera, leptospirosis, acne, and even some protozoa. So it works fairly well, but it does cause some issues with your commensals, or can cause some issues with your commensals. Chlorampphenicols, these bind to the larger subunit, or the 50S subunit of the ribosome, which is here. You can see this little X on the large unit. And this prevents peptide bond formation. So by preventing peptide bond formation, you're not going to form a nice long chain, block essentially elongation of the amino acid chain or peptide. And this can be very toxic to the cell. One drawback to using chlorampphenicols is they tend to cause aplastic anemias in a small percentage, so about 0.1% of individuals who take the drug. But once this happens, it is irreversible. And an aplastic anemia is essentially a reduction in red blood cells, and this is due to the change or shape of the red blood cell. And when the red blood cell is changed, or when its shape is changed, it can no longer bind oxygen properly, so it cannot transmit oxygen throughout the body. And this can be a pretty serious problem. So again, we don't use chlorampphenicols terribly often unless there's no other option left, just because of this problem. Erythromycins, So this has a large lactone ring with sugars attached. It's relatively broad spectrum with low cytotoxicity, and this blocks protein synthesis by attaching to the ribosome. Works well against mycoplasmas, legionnosis, chlamydia infections, pertussis, diphtheria. It's becoming kind of the mainstay drug for a lot of treatments, just because it is pretty easy to take, and it lasts in the body for fairly long periods of time. Clindamycin, it's broad spectrum, derived from lindamycin, which is a product produced by a fungi, and it can cause adverse reactions in the gastrointestinal tract because it is broad spectrum, so its applications are limited. One of its big applications is actually an acne treatment, and it can be used as a topical acne drug because it targets all microbes that could potentially cause the acne. And since it's on the skin, you don't really care if it kills some commensals. This is what they look like. So here's a tetracycline, here's a erythromycin. Oxazolidinones, this essentially bind to the entire ribosome, and this prevents initiation. So it just stops the ribosome from actually binding the mRNA. So as you can imagine, this is pretty effective because you get absolutely no translation whatsoever. The big drug in this group is linezolid, and this is one of the few new antibiotics that's kind of on the market. It is completely synthetic, so it is extremely expensive. One pill of this drug is around $150 to $200, but it does work extremely well with fairly low cytotoxicity, and it's used primarily when you have no other options. So if you have an organism that's causing disease that is resistant to almost everything else you throw at it, your best option is to use linezolid. Most doctors kind of keep this drug close to their chest and do not use it willy-nilly so that resistance doesn't develop. And we'll talk about in a little bit how resistance can develop if you use the drug inappropriately. And the last class of drugs we're going to talk about for prokaryotes is drugs that target folic acid synthesis. And these are primarily sulfa drugs. So these are 3 different flavors of sulfa drugs: sulfonamides, trimethoprim, and sulfones. And all of these essentially do the same thing, and this is blocking the production of folic acid by essentially using up the enzyme required for folic acid production. These drugs work. They tend to not work very broad spectrumly or very effectively, so we only use them in pretty specific cases, and they often can have fairly high cytotoxicity associated with them, so we tend to just use them in pretty severe cases. So again, sulfa drugs primarily will bind to the active site of the enzyme required for folic acid synthesis. And this uses the enzyme or preoccupies the enzyme so that it can no longer produce folic acid. Alright, so now we'll switch gears a little bit and get away from prokaryotes and switch over to the eukaryotic side of things. The first group in the eukaryotes we're going to talk about are fungal infections. So remember, fungal cells are eukaryotic, so it can be a little bit difficult to treat just because they are very similar to our own cells, since we are eukaryotic as well. But there are some, pretty distinct dissimilarities that we can target. The first one being eugesterol, which we talked about in, I guess, a couple weeks ago when we were talking about eukaryotic pathogens. Remember, eugesterol is a molecule found in the plasma membrane of fungal cells. It is a sterol that is unique to only fungus, so we can target it while leaving our cells alone. And another potential target would be chitin. And remember, chitin is a molecule found in the cell wall of fungi. Most drugs don't target chitin, however, and this is because eukaryotes do not need to constantly produce their cell wall. Once they produce it once, they have it for quite a long period of time. So it doesn't work very well with organisms that are already established, although it will prevent the growth of new organisms. And there are 4 main groups: macrolide, polyene antibiotics, gristofluvins, synthetic azoles, or flucostines. So we'll first talk about some of the most widely used antifungal drugs, and these are the polines. And these will bind to the membrane of fungal drugs, and this is primarily to eugestrol. However, eugestrol looks fairly similar to cholesterol, which is the sterol found in our plasma membranes. So these drugs tend to kind of cross-react or also react with our cells. So they can be very, very toxic, especially if given intravenously. If given orally, they tend to be very mild toxicity-wise, but once they get into the bloodstream, they can cause some severe problems. So most of the time, these drugs are only used in really severe antifungal cases, and these are, severe fungal cases. These cases tend to be fungus in the blood or fungaremia. And again, we've talked about this before, where fungi primarily only infect individuals who have a severely weakened immune system. So you really only see this in people who have AIDS or cancer or something that will really limit the properties of the immune system. The big group, our drug here is amphotericin B. It works really well, but it does have a very high cytotoxicity. It's probably one of the worst drugs when it comes to cytotoxicity, especially when administered intravenously. But if you have a systemic fungal infection or fungaremia, you're in really, really bad shape. Chances are it's going to kill you, so you have to do whatever is possible. So taking amphotericin B is your really last resort or only option. And it does work, but it's a long shot, and it typically causes some really nasty side effects which can kill you as well. So, in a nutshell, amphotericin B is a potent drug, but it can also be pretty toxic to our cells as well. Azoles, so this is probably the most common antifungal class, and this inhibits eugestrol synthesis, and this tends to have a very low cytotoxicity to our own cells, and that's because you're targeting an enzyme within the cell that is only found in fungi because it is used for production of eugestrol, which we do not have. So it works pretty well. Most of the antifungal drugs that you think about, like miconazole, for example, which is primarily used for yeast infections, or even athlete's foot, is an azole. Works well, low cytotoxicity, so it's a pretty good option. Gristofluvins, this works pretty well against ringworm, and this inhibits microtubules. So microtubules are produced primarily in cells that are actively dividing. This occurs, or they're produced in very high numbers when a cell is going through mitosis. And since most of our cells in our body, with the exception of our hair and a couple other cell types, primarily in the immune system, our cells are not dividing. After puberty, our cells are pretty much established and are with us for the rest of our lives. So if you take this drug, it works pretty well at targeting the actively dividing cells or the potential pathogens or eukaryotic pathogens, but leaving our cells alone. So synthetic azoles, so you have ketoconazole, fluconazole, clomidazole, miconazole. These are very broad spectrum antifungal agents. They work pretty well both orally and topically. You can use them for many different types of mycoses or fungal infections. So things like vaginal fungal infections or yeast infections, oral candidiasis or yeast infection of the mouth, and some systemic mycoses. However, they don't work very well in the blood just because they tend to get degraded fairly easily. Fluconazole can work in AIDS-related mycoses, but it doesn't work nearly as well with amphotericin B. In fact, if you treat with fluconazole, you usually treat with amphotericin B at the same time. Flucostines, this is an analog of a nucleotide, primarily, or a nucleotide of cysteine. What this does is it prevents DNA replication, and since again most of our cells are done actively dividing, they no longer need to replicate their DNA, so it targets only the eukaryotic cells that are actively dividing, which are primarily the fungal pathogen. Can be useful in treating certain cutaneous mycoses, and again, this is because it can cause some other nasty side effects if taken orally, primarily with loss of hair, because your hair is constantly producing or constantly dividing. And usually it is combined with amphotericin B for systemic mycoses, and this combination is very, very, very toxic. But if you have a very severe systemic mycosis or fungeremia, you really have no other option. Antihelminthic drugs, so these primarily target microtubule formation, and it's again because helminths tend to be actively dividing always, and since our cells are not, if we target their actively dividing nature or their microtubule production, we can kill them. Most of them work very well at blocking replication, but it does not work fairly well at killing adults. There are a few that will, but so what this means is you tend to have to take the drug for long periods of time until the adults die naturally. Most effective drugs immobilize, disintegrate, or inhibit the metabolism of all stages of the life cycle, but again, this is fairly difficult to do. Mebendazole, Thiabendazole, and ivermectin are the most common flavors of anti-helminthic drugs. They tend to be fairly broad spectrum, and this is because helminths are large multicellular eukaryotes, which means they do not mutate fairly rapidly, and they all have fairly conserved mechanisms of replication and growth. So if we target one group, we often can target many groups, and since they do not mutate rapidly, there's very low resistance found with these drugs. So there's only about 5 or 6 antihelminthic drugs available, and they all work fairly well. Antiviral chemotherapeutic agents, so this is fairly difficult group of antimicrobials to develop. And this is primarily because viruses will take over your own cells and use the cellular machinery for their own benefit. So it becomes very difficult to find selective targets in this case because they are using your own cells and your own cellular machinery, which looks very similar to the cellular machinery of an uninfected cell. So if you try to target one, you'll probably target the uninfected cell as well. So toxicity tends to be an issue with a lot of these antiviral chemotherapeutic agents. However, there are ways of targeting just the viral life cycle itself. Often these require a lot of research and are very expensive to develop. So for that reason, pharmaceutical companies have put a lot of money into antiviral chemotherapeutic agents because they can charge very high price tags for these. So it makes them a huge profit. So most new antimicrobials that are coming out in the market are actually antiviral in origin, and they're usually antiretrovirals that are primarily used to target HIV replication. So 3 major modes of action for these antivirals are preventing penetration of the virus, so stopping the virus from actually getting into the cell of interest, blocking the transcription and translation of the viral molecule, and preventing the maturation of the viral particles, so they can no longer be released and infect a new unsuspecting cell. Some antivirals, amantadine, so this inhibits the replication of influenza A virus. Acyclovir, which is a very well-known antiviral drug. This disrupts herpes viral replication. It is a very, very nicely designed drug. It took a lot of energy. The guy who actually developed it won the Nobel Prize for doing this. Essentially, it is a glucoside analog, or nucleoside analog. And what this does is it prevents the replication of herpes viral DNA, but not human DNA. So it's very, very, very selective. So for that reason, it has almost no cytotoxicity associated with it, or nasty side effects. Also, acyclovir is what we call a prodrug. So that means when you take it and take it orally and it gets into your gut, is actually in an activated form. The only way it becomes activated is when it's actually inside of a cell that it is targeting. So this also greatly reduces the amount of side effects because while it's swimming around in your blood, it isn't in its active form causing any kind of damage to your cells. And ribavirin, this blocks RNA synthesis primarily in influenza. So some of the mechanisms involved in antiviral agents. So you can actually inhibit the virus from entering. This is called a fusion inhibitor. So here we have HIV. HIV needs CD4 as well as CCR5 or CXCR4 to enter the cell. If you block the CCR5, primarily by using a CCR5 blocker, you stop the virus from getting in. This stops the life cycle. You can also do this with influenza, primarily by blocking hemagglutinin. Remember, hemagglutinin is an enzyme found on the surface of influenza, and this is used for viral entry. You can inhibit nucleic acid synthesis, so you can have things like acyclovir, which we talked about before, which physically targets antiviral or viral DNA, preventing the virus from producing new DNA, thus preventing its replication. But there are many other flavors of this. You can also have an HIV nucleotide or non-nucleotide analog inhibitor. And these essentially block reverse transcription. Reverse transcription is where HIV takes its RNA, turns it into DNA, and without DNA it can't produce the rest of its viral components. And then the last group is an inhibition of viral assembly or release. And this is what we call a protease inhibitor, especially with HIV. And this prevents the HIV proteins from being cleaved, and if they're not cleaved, they are no longer functioning. So if they're not functioning, the cell or the virus cannot reproduce. Interferon, so this is kind of a natural way of targeting viruses. And an interferon is essentially a chemical produced by your immune system, or actually all cells in the body to a certain degree. And it is what we call a cytokine. So when this interferon is released from a cell, it will bind to neighboring cells, and it warns the cell that there is a virus present. So interferon is only released when a cell is infected with a virus. So it becomes infected, releases interferon. This interferon is released into the rest of the body, and this tells tissues and cells in nearby proximity that, hey, there's a virus around, I'm infected, get ready. So it allows the cell to kind of prepare itself for the viral infection, turn on natural antiviral mechanisms, and this will stop the viral replication to a certain degree. It does work fairly well. The most headway made with interferon treatment is actually with hepatitis C. It's kind of the main way of treating hepatitis C, which is a chronic viral infection that targets the liver. Typically you treat with interferon as well as efavirenz, which is an antiviral drug. And this will completely or slow down the replication of the herpes virus, thus prolonging the life of the individual who has the hepatitis C infection. But you can get some serious side effects because it is a cytokine, and cytokines are very, very potent. You need to use it in very small doses. And not everyone responds to the same dose, so it takes a lot of trial and error before you can actually develop the right regime or treatment. Alright, so now we'll talk about what happens when microbes are in the presence of these drugs and how they can eventually develop something called drug resistance to these drugs. And this allows the organism to grow in the drug, and when it's growing in the drug, it's not affected by the drug, and thus can cause infection still. So essentially, drug resistance, this is an adaptive response in which microorganisms begin to tolerate an amount of drug that would ordinarily be inhibitory, and this is essentially natural selection at work. What this means is that when the organism is in the presence of this drug, randomly there's bound to be some mutations that can occur in these organisms as they are dividing. Sometimes these mutations will actually give the organism a benefit, and this benefit would be the ability to live in that drug. So if it can survive in that drug, it now has a growth advantage, and it eventually will take over the entire population. And now you're stuck with a whole bunch of organisms no longer responding to the drug, which can cause some serious infections that can be difficult to treat. Now this can be intrinsic or acquired. Intrinsic means it's a mutation that it develops on its own, where acquired would be something that it picks up from its neighbor. And this is via conjugation, transformation, or transduction. All of these processes we talked about earlier on. And microbes tend to become newly resistant to a drug after spontaneous mutations, so this is the intrinsic resistance, or acquisition of the new drug from an R factor or a plasmid that has some sort of resistance gene associated with it, and this is the acquired drug resistance. Oops. So resistance mechanisms. So antibiotics are present in nature. These are produced by organisms to kill other organisms. So it is just a natural phenomenon for prokaryotes to develop some sort of way to get around this. And again, this is primarily by having mutations that either allow them to produce something like an enzyme that will break down the drug or actually block the drug from doing its job. And they are pretty capable at doing this, and they can do this fairly quickly. And this is because they divide very, very rapidly, especially when you're dealing with things like Staphylococcus aureus, for example. At ideal conditions, Staphylococcus aureus can divide every 15 to 30 minutes. So that means every 15 or 30 minutes, you're essentially opening up the possibility for a mutation to occur. And if that mutation occurs in a position that allows for this organism to grow in an antibiotic, you now have developed an organism that can survive in your antibiotic, and it will quickly take over the population, making it very difficult to treat. Drug resistance has arisen for all antibiotics, and there are some ways to prevent this, and we'll talk about those in a little bit. And two main strategies are to prevent access of the drug, so preventing it from getting to the actual site where the drug does its job, or altering the drug by cleaving it with an enzyme. So here we have drug inactivation. This is using something like a penicillinase, which cleaves the beta-lactam ring, which you can see here, and making it ineffective. So this is physically changing the drug within the bacteria so it no longer functions. You can prevent the drug from actually getting in. So most drugs require some sort of cellular receptor found on the surface of the bacteria to physically enter the bacteria. And if you change this surface so that the drug can no longer get in, then the drug can't and you survive. You can actively pump the drug out. So say if you still let the drug get in through these receptors, you can now produce something called a drug pump, or another flavor of a receptor that will actually take the drug as soon as it gets in and pump it back out so it can no longer work within the cell. You can change the binding site. So this happens very frequently in a lot of the protein inhibitors, or the translation inhibitors we talked about earlier. Essentially, the organism will slightly change its ribosome so that the drug no longer targets, thus preventing the drug from functioning. Or if you're talking about sulfa drugs, you can switch to a totally different metabolic pathway, which means instead of using that enzyme that was required to produce folic acid before, you just switch to a different enzyme that the drug no longer works against. And what tends to happen is with antibiotic resistance is you have organism, which you plate on a plate. Let's say this plate has ampicillin in it. And as you can see, most of these organisms turn black, or that means they die. And that is because they do not have the ability to grow in ampicillin. But a few of these guys who are brown here have the ability to grow in ampicillin because they have some sort of genetic resistance to ampicillin. They probably produce something called a beta-lactamase, or an enzyme that breaks down the ampicillin or antibiotic. Now, as these organisms grow or sit on the plate, they will start to grow and divide, and as they grow and divide, they produce new organisms that essentially have the same ability to grow in this ampicillin. So you can see here they start to take over the population because obviously they're the only ones who can grow. And by the end of the day, you're left with a whole bunch of organism that is now resistant to your antibiotic. And they quickly take over the population because they have a significant growth advantage. So this is a really, really important slide, especially for clinicians. One problem we have in medicine today is that antibiotics tend to be administered inappropriately. If you administer them inappropriately, especially in a very large population, you're just increasing the chances or likelihood of drug resistance occurring. So there are some things we can do to prevent this drug resistance from occurring. And the big one is to limit the drug use. So if you limit the drug use, you limit the exposure of organisms to that drug, so there's less selective pressure. There's less organisms coming into contact with the drug. There's less organisms mutating to try to survive in the drug, thus you get less resistance to the drug. But it's also important to use the drug properly. So this is using your full dose and making sure that you use the dose for the amount of time required. So most of you, I'm sure, have taken amoxicillin before, unless you're allergic to it. And amoxicillin is primarily used for treating things like strep throat, so Streptococcus pyogenes. And what this drug works extremely effectively, but it has a relatively short half-life. So what tends to happen is that you need to take the drug for around 28 days. And this usually requires you taking the drug twice a day. And as you can imagine, this is pretty annoying. Most people don't want to take a drug for 28 days. They don't want to take it twice, especially big horse pills, which amoxicillin usually looks like. And what tends to happen is after you take the drug for a couple days, let's say a week, you start to feel better and you don't feel like you're sick anymore. So most people will say, well, what the hell, I don't need to take the drug, it's useless. But what tends to happen is during those first 7 days of you taking the drug, you actually kill probably 99% of the organism. So that's why you start to feel better. But note that there's still 1% of the organism left. And that 1% of the organism that is left happens to be the 1% of organism that is most resistant to the drug. That is why it survived through those 7 days of the drug treatment. So now if you decide to go off of the drug after 7 days, those resistant 1% can now start to grow and divide. And then you start to feel sick again. So then you're like, well, I'll just take my amoxicillin. But now you have an infection with Streptococcus pyogenes that is slightly resistant to amoxicillin, and the amoxicillin no longer works as effectively. So maybe now it takes 14 days before it starts to work. So this is why it's really important to take your drugs for the entire prescribed time. So if you were to take the drug instead of 7 days for 28 days, those resistant bugs would be in the drug for long enough to where eventually it would wear them down and they would probably die. So this would greatly reduce the likelihood of resistance occurring. It's also important to use something like narrow-range antibiotics. And the reason why this is nice is because if you use narrow-range antibiotics, you're only targeting the organism of interest and not your commensals. And since your commensals are primarily there to take up space and prevent pathogenic organisms from growing, if you can target the pathogens and leave your commensals alone, you greatly increase the chances of clearing the infection and reducing resistance. Also, multiple drug treatments. So taking many drugs at once, especially for pretty resistant bugs, can work fairly well because it's much harder for the organism to develop resistance to multiple different pathways or multiple different maybe enzymes or targets of the drug. Whereas if you're just taking one class of drug or one type of drug, it's fairly easy for the organism to develop resistance to it. Some interactions with the drug and the host. So these are the adverse reactions, or what we call cytotoxicities. So obviously you can get toxicity to organs, especially if you have a drug that kind of cross-reacts with your cells as well as the target organism. You can also get allergies, which is much more common in antibiotics or drugs that are produced from natural sources. And again, disruption of normal flora or killing your commensals. Without your commensals, you essentially open your body up as a free breeding ground for potential pathogens, and you can also disrupt the digestive tract. But there are also other adverse reactions, such as this. This is what we call a drug eruption, and this is a skin reaction to the drug. It can be fairly mild, such in this case, which just looks like a nasty rash, and as soon as you take the drug or stop taking the drug, these kind of things will go away. Or it can be very serious, like Stevens-Johnson syndrome, where essentially you have desquamation of the endothelial cell or epithelial cells. So your skin basically kind of sloughs off, and it can be very, very serious with about a 50% mortality rate. So this is something you have to keep in mind when administering drugs. You don't only want to target the organism, but you want to target the organism but leave your cells alone. So how do you select antimicrobial drugs. So the primary way is by doing susceptibility testing. And this is testing the organism that's causing the infection and seeing what type of antibiotic is effective against it. And this becomes extremely important if you're treating with an antibiotic and it's not working. And this means the organism is probably resistant to it, so you should probably pull out a clinical sample of this, isolate it, and try to determine the most appropriate antibiotic to use. And then you can develop something called a therapeutic index, and the therapeutic index is essentially a drug that targets the organism but has low cytotoxicity in the patient. So one way of doing an antibiotic susceptibility test is something called a disc diffusion assay or Kirby-Bauer test. We will physically do this in the lab. And essentially what this is you take your organism of interest and you plate it on a plate so that it completely covers the plate or causes a lawn of bacteria. That's what you can see here in green. You then take antibiotic discs, which are little paper discs that are coated in a specific antibiotic at a specific concentration, and you place this on top of the media. You then incubate overnight and the organism will grow. Now, if the organism is susceptible or reacts to the antibiotic and the antibiotic actually kills it, you will see something called a zone of inhibition, which is a clearing around the disc. Now, the larger the zone of inhibition, the more susceptible the organism is to the antibiotic. And the reasoning for this is that when you place the antibiotic on the disc, the antibiotic will actually diffuse through the media. And as it diffuses, it goes from higher concentration at the disc to a lower concentration further away from the disc. So what this means is that if you have no growth pretty far away from the disc, that means the organism's not growing at this very low concentration, which means it's really, really susceptible to the antibiotic. Conversely, if the organism can grow right up to the disc or shortly away from the disc, it means it can grow in relatively high concentrations of the drug, and this means it's probably fairly resistant to it. And you can take these zones of inhibitions and measure them in millimeters and compare them to charts, and it will tell you how resistant or susceptible the organism is. So it works pretty well. Biggest drawback to this is you really don't know what the concentration of drug is. So yes, you know it's lower here, but you don't know exactly how low it is. So you don't know how many milligrams or grams or whatever the case may be of the drug is present. So a way to get around that or to do a susceptibility test but also test for the concentration of the drug that is most effective is by using something called an E-test strip. And an E-test strip is very similar to the Kirby-Bauer. You essentially plate a lawn of bacteria, but instead of putting a disc on the plate, you place an E-test strip. And this is essentially a strip of the antibiotic that has a concentration gradient on it. So you have a very low concentration at the bottom and you have a very high concentration at the top. You place this and you'll get something that a zone of inhibition that looks kind of like this weird teardrop shape, and you can go down the teardrop shape until right where the organism starts to grow, and that will tell you the concentration or the minimum inhibitory concentration or the MIC for this drug of interest. So here you can see with this IP antibiotic, you can see its MIC is about a 1. And this tells you that using 1 gram or milligram, whatever this is, it doesn't say, will be very effective at killing the organism. So that's the amount that you want to give to your patient and try to make sure is in the blood at all times. And you can see this organism, or with this antibiotic, it's pretty effective at 1 as well, but you can see this zone of inhibition is much larger. So that tells you that this organism is probably more resistant to this drug, or more susceptible to this drug, than this drug. Another way to do this is the tube dilution assay, which is a lot more time consuming, but it's much cheaper. Test strips tend to be pretty expensive. And this essentially is doing a serial dilution of your antibiotic, not of the organism, where you start off with a high concentration, and then you dilute it so that you get a lower concentration of the antibiotic, and then you place the same amount of organism in each tube, and then you look for growth the next day. So here you can see the organism grew here and here, but it stopped here. So this tells me the minimum inhibitory concentration is actually 3.2 micrograms per mil. So that's the concentration of drug that I would try to use. You don't want to go too low because it could cause issues, and you don't, or it won't work properly and develop resistance in the bacteria. And you don't want to go too high because it can actually kill the organism or hurt yourself as well. And with all this information, you develop the therapeutic index. And the therapeutic index is a way of determining the best drug to use, so it's most effective at targeting the organism, but also it's not very toxic to your cells or has very low cytotoxicity. So it's the lowest risk of side effects versus the highest probability of killing the pathogen. And it's essentially a ratio between the toxicity to the organ or to yourself and the effectiveness against the organism. So here we have 50 micrograms of the antibiotic is toxic to us, but 5 micrograms is effective. So this gives us a TI of 10. But if it was 50 micrograms is toxic and 1 microgram is effective, you have a TI of 50. And a TI of 50 is better, or a higher TI is better, because this means you have a better or larger window of where the drug is effective but not toxic to our cells. Alright, and that's pretty much it. So hopefully this was a good lecture. And if you have any questions on any of this information, please ask me on either Wednesday or Thursday, and we can talk about it in person. And I am going to finish grading your exams tonight, and I will post them very shortly. Okay? Alright, well, have a good time off, and I will see you guys soon.